Shiri Sudri, sorry, from the Rambam Healthcare Campus. She's going to talk to us about visual loss in children. In with the new.
Thank you. Okay, bring it up. Okay, so good morning, everybody. First, Udi on the organizing committee. Thank you so much for giving me the pleasure and privilege of being here. Certainly an honor. I do realize that almost the last barrier between everyone is here, including myself, and lunch. So I promise I'll try to be as clear, as precise, and as timed as possible. So the structure of the eye is very similar to a camera, with the anterior segment of the eye serving as the optical component, whereas the retina lining the back part of the eye is the light-sensitive tissue responsible for phototransduction, or the process of transformation of light energy into electrical neuronal activity. The retina is actually a direct extension of the brain, and normally it has a multi-layered structure with several layers of neurons that are interconnected by plexiform layers of synapses. Underneath the retina are the retinal pigment epithelial cells, and in the RPE, together with the photoreceptors, is the compartment where the phototransduction actually takes place. The center of the retina, or the macula, is also called the center of vision. Now, we all know that normal vision involves many aspects, such as daytime vision versus nighttime vision, color vision, contrast sensitivity, and others. Proper macular function is crucial for many of these functions, and therefore crucial for our well-being. So how does the macula do that? So there are two types of retinal photoreceptors, with the rod photoreceptors mediating our nighttime vision, whereas the larger cone photoreceptors mediate our daytime vision, our color vision, and the acuity of vision. The macula has a high concentration of cones, with its very center being a cone-only area, thus providing us with the sharpest vision. Retinal degenerations is a group of diseases that are characterized by progressive retinal cell death that can be caused either by inborn genetic variations or can be acquired later in life. Many of these degenerations present early during the first decades of life, sometimes even during childhood, when the first symptoms of progressive visual decline begin to appear. It is easy to imagine the devastating impact that such degenerations may have on our well-being. Many of these degenerations are characterized by typical phenotypes of degenerated retinal cells. And where many retinal cells can be involved, we know that many of the retinal degenerations primarily affect the photoreceptors and the RPE. And therefore, currently, proper classification of these conditions is based on genotype mapping. Perhaps you can also notice that, as I present here in the slide, that many of these conditions are characterized by the appearance of abnormal tissue deposits collecting within the retina. Now, our research group is fortunate enough to be collaborating with Udi's group, and our research interest is focused on better understanding of the molecular nature and the biological consequences of these deposits. Today, however, I chose not to expand on this fascinating subject, but instead to share with you a broader view on the evolving field of retinal degenerations. So I would like to highlight a few of the recent advancements in the clinical and laboratory aspects of the diagnosis and management of retinal degenerations. And let's start with the diagnostics. So adaptive optics is a technology that has revolutionized the way we can see the retina. The main limiting factor for in vivo retinal imaging is high-order optical aberrations that normally exist in the eye of each and every one of us. Adaptive optics can overcome these limitations by using a wavefront sensor and a specialized deformable mirror, thus achieving a lateral resolution on the order of 2 microns that enables to visualize individual retinal cells that are otherwise not visible with standard imaging modalities, such as the photoreceptors and the RPE. So here's a normal retina, now imaged with SLO, which is a conventional imaging method. Now adding adaptive optics to the loop. Each of the white spots that we're seeing represents light that is being reflected from an individual cone photoreceptor. When averaging several frames like that together, we get a higher signal-to-noise ratio. If we combine several Oslo images, adaptive optics images together, we get a montage that enables correlation with macroscopic clinical findings. Here are a few examples of cone photoreceptor mosaics that were imaged in vivo in a normal eye, shown side by side with images of photoreceptors that were obtained from human retinal hole mounts. And I think it is easy to appreciate the striking similarity between those images that were obtained in vivo and ex vivo. Direct visualization of cones also enables quantitative analysis of cone spacing, or the average distance between photoreceptors, which is a robust measure of the integrity of the photoreceptor mosaic in that region. We know that cone spacing measures correlate favorably with normative data that were obtained from histological studies of normal eyes. So with that in mind, it's easy to imagine the utility adaptive optics imaging has shown in clinic, research, and in clinical trials of retinal degenerations. So let's look at a few examples. Here's a young patient with retinitis pigmentosa, a retinal degeneration characterized by progressive degeneration of frauds, eventually followed by cone loss. Perhaps you can appreciate the widespread atrophy of the photoreceptors in the RPE around the macular center. Adaptive optics imaging shows remaining photoreceptors in the very center, whereas in adjacent retinal areas, the photoreceptors have degenerated and we're actually seeing surviving RPE cells. In this condition, the degenerative process proceeds from the periphery of the retina towards the center in a ring-like fashion. So repeated imaging enables actually longitudinal tracking of the disease progression with high sensitivity. Here's another patient with Best maculopathy showing a typical vitelliform lesion at the macular center. These yolk-like lesions are caused by accumulation of lipofuscin, which is a compound byproduct of the visual cycle within RPE cells. We know that lipofuscin is toxic to the RPE cells, yet in these subjects, it's only the central vision that eventually becomes impaired. Adaptive optics, again, may show some insight, and indeed shows increased cone spacing values directly over the vitelliform lesion, but normal around it. Here's another patient with similar findings, and again, in a third patient, increased cone spacing measures directly over the vitelliform lesion, but normal around it, indicating that chronic accumulation of lipofuscin may be toxic to the overlying photoreceptors, perhaps suggesting that efforts to reduce lipofuscin deposition may improve the cone photoreceptor survival in these eyes. So beyond doubt, adaptive optics has improved our understanding of the cellular anatomy and biology in retinal degenerations. An increasing number of clinical trials are currently employing this imaging modality for patient selection, stratification, as well as for monitoring of treatment outcomes. And we certainly expect that the use of such imaging systems will continue to rise in the future. Now, in terms of treatment, novel treatments for retinal degenerations, a series of interventions offer potential opportunities in different stages of the diseases. The eye, if you think about it, may represent an ideal target for gene or cell therapy. It is conveniently accessible, it is easily imaged, it is small, thus requiring low volumes for injection. It permits specific targeting of intraocular tissues with the blood-ocular barrier actually minimizing systemic exposure. The retinal cells normally do not divide, which means that sustained transgene delivery is actually possible. And most importantly, perhaps, the contralateral eye can serve as an internal control, thus allowing for accurate assessment of the therapeutic outcome. Viral vectors are among the most common approach in gene therapy, with adeno-associated virus, thus far being the safest and therefore the broadly preferred vector. Subretinal injection can deliver those vectors in proximity to the photoreceptors and the RPE, and indeed has been used in most gene therapy studies for retinal degenerations. encouraging animal studies provided results that paved the way towards ongoing clinical trials. The first success in humans was seen with the RPE65-associated Leber congenital amaurosis, which is a very severe form of retinal degeneration characterized by visual impairment from birth. Subjects with the disease received a single subretinal injection of the AAV2 vector carrying the RPE65 gene in their worst affected eye. Following the injection, continued improved in visual function, including ambulatory vision and light sensitivity, were noted through at least 3 years. Based on these results, the first retinal gene therapy, Luxturna, became available after receiving FDA approval last year, and as a matter of fact was also approved by the European agency approximately a week ago. Additional treatments for additional retinal degenerations are also being currently investigated in clinical trials. For example, Stargardt's disease is the most common form of inherited macular dystrophy and the most common cause for severe vision loss, central vision loss among young adults. Proof of concept studies in knockout mice showed that delivery of the underlying human ABCA4 gene was associated with significant improvement in retinal function and again paved the way towards an ongoing clinical trial. In all these cases, the goal is to treat early, to try and stop the degeneration and try and save as much vision and as much photoreceptor structure and function as possible. When the disease severity is too profound and when photoreceptor degeneration is too extensive, alternative approaches may be required for restoration of vision. Such experimental approaches include retinal prosthesis or optogenetics, which is a method to genetically introduce a light-sensitive protein into the light-insensitive cells of the retina, thus potentially transforming them into artificial photoreceptors. This method again showed success in preclinical studies and is being moved towards a clinical trial in the present. Lastly, stem cell therapies offer an alternative approach for retinal degenerations, especially in cases when cellular degeneration and loss is profound. One of the main advantages of cell therapies is that they are mutation independent and therefore can be employed in a wide range of conditions. Naturally, for retinal degenerations, the cell sources that are being explored to the maximal extent are the photoreceptors and the RPE. Presently, there are at least 15 ongoing clinical trials looking at stem cells for retinal degenerations. One of them actually is being performed in Jerusalem by CellCure, with my dear friend and esteemed colleague, Professor Eyal Banin, being the leading scientist. So the results of some of these trials are expected with much enthusiasm over the near future. So just to summarize, these and other novel treatments, we're all hopeful that offer a real opportunity for potentially curing the previously incurable retinal degenerations. It is broadly agreed that in order to keep moving this fascinating field forward, a strong collaboration, strong collaborative efforts are required among academia, industry, and funding agencies. And with that in mind, we are all hopeful that this will be the case in the future, and we look forward to see what the near future brings for our patients and their families. So with that, I thank you for your attention, and I'm happy to take questions. Thank you. Yes. Thank you. So for AMD, certainly one of the most fascinating and painful dilemmas that we all encounter on a daily basis. We are not quite sure about the exact pathophysiological mechanism of AMD yet. I mean, there are many suggestions, but none is really a solid consensus. We think that likely it is not necessarily a purely genetic condition. We think that there might be some genetic predisposition, but perhaps it is also has a strong acquired foundation which is being explored. We actually are also exploring one of the an interesting hypothesis on the cellular damage that occurs in AMD. But we don't necessarily think that the main factor is a genetic one. As for VEGF inhibition, VEGF likely plays a role in a specific phenotype of the disease, which is the neovascular form of the disease, that is being targeted either by antibodies or through additional approaches that are also becoming more and more feasible nowadays. But I would say that although perhaps the final outcome, which is cellular degeneration and loss of photoreceptors and RPE, is similar among AMD and those inborn errors of retinal physiology, you may call, not necessarily the mechanism is the same.
Thank you very much.
Thank you.